Hi everyone, my name is Katie Shear, and today I will be presenting on the dynamic culture of trabecular meshwork cells on biomimetic 3D collagen scaffolds as a part of Dr. Melissa Krebs' research lab. So just some background on glaucoma, which is the disease being studied. Glaucoma is the leading cause of irreversible blindness, and as of 2012, it was diagnosed in over 4 million people in the US, which is a number expected to double by the year 2050. The cause of glaucoma is attributed to an increase in the intraocular pressure of the eye, which puts pressure on the optic nerve, damaging it and causing the blindness. This increase in pressure can be affected by the flow of aqueous humor in the front of the eye through the trabecular meshwork, which is a native tissue at the base of the cornea and is responsible for draining the flow of this aqueous humor. So in order to go about studying this, we designed a model trabecular meshwork tissue, here referred to as TM, in vitro. This was done by creating a collagen scaffold where a collagen slurry was made with added glycosaminoglycan components, which were native in the TM tissue in the eye naturally and are an important part of the structure. This slurry was then put through freeze casting, where it was either flash frozen or done at -80 degrees. So either FF or -80 from here on. And then this was either with non-aligned pores or aligned pores, which was done through the presence of a metal plate at the base for aligned or not. And this allowed for different pore sizes and structural compositions. The seating of these scaffolds with the TM cells made for a very good working tissue model in a 2D static method, but we wanted to see if we could make that better. So after that, this is just showing the visuals of the 4 different freezing types, with the non-aligned referred to as NA and aligned as A in the future. And so the part of the research that I've been really active on is the part with 3D dynamic perfusion chamber, where we're trying to take it from the 2D static to the 3D dynamic model. So this is, as you can see in the picture, a series of 12 interlocking chambers with the seated scaffolds placed inside, which is all done in a sterile environment. A continuous flow of oxygenated media is then pumped through the system, and it is allowed to run for the desired time, typically 48 or 72 hours, and then the scaffolds are removed, and the cell proliferation and gene expression is then studied on these cells in a quantitative and qualitative manner. So this is data taken from one of our successful runs so far this semester. As you can see, a cell proliferation study was done in a quantitative means, so that you can see the 4 different types of freezing, NF, AF, N80, and A80. And this is shown that all 4 freezing types had cells present and growing successfully in relation to the control of the absorption test. So then the successfully grown cells were taken to confocal imaging, where you can see in these 4 pictures each of the 4 different types of scaffolds, and the small blue dots are the surviving cells which successfully grew. So what this tells us is that the proliferation studies demonstrate the success of the perfusion chamber in modeling the human trabecular meshwork, which is a massive improvement because this will allow for the next steps of drug testing for the disease of glaucoma. The entire hope with making this a 3D dynamic model was to make it as realistic and as good of a representation as possible to the native eye. And so this will then allow us to test drugs and how they affect protein expression, the structure of the trabecular meshwork, including the extracellular matrix outside of the cells, and also the intraocular pressure. And so our long-term goal with this is to be able to understand drugs and how they will impact these things, and hopefully be able to treat glaucoma before it is a problem and make this a far less dangerous disease. Thank you.